So let me show you on paper how the RT-PCR test really works to detect an RNA molecule. So in this case, it's going to be the COVID virus RNA, and that would be extracted with RNA from a tissue sample. It could be somebody's blood sample, and there'll be a mixture of RNAs in there that RNAs are transcribed from the human genes in the cells, but then there's the viral RNA in there. And all the RNA goes through an RT-PCR reaction, and in this case the RT will stand for reverse transcriptase. And so there's an enzyme called reverse transcriptase, and what it does is it will use RNA as a template, and with a primer it will make a copy of the other strand version, and it will be using DNA. So it's called copy DNA or cDNA. And the normal base pairing between RNA or DNA bases are used, and you'll end up with a DNA molecule. Once you have DNA, the rest of the RT-PCR reaction that you want to use is the PCR part of the reaction, and that's called polymerase chain reaction. And how that works is once you have a copy of this DNA, which now represents sequence unique to the virus, and there's also DNA in there, cDNA, that's been made from RNAs that were from the human cells. But if you use what are called primers, you can basically have little pieces of DNA that are called primers, and they will do the normal base pairing to one strand. Now there's another primer in there which isn't going to be used yet because we just have one strand of cDNA. But what happens in the first cycle of the PCR is basically a thermostable polymerase called a Taq polymerase enzyme will make the second strand of the DNA. And this is all base paired. Hydrogen bonding between the bases. You know that A pairs with T and G pairs with C. So now I've gone from a cDNA, and I will turn this into what is now a double-stranded DNA. DS, double-stranded DNA. The next cycles of PCR, 2, 3, and 4, etc., what happens is you heat up this DNA to like 95 degrees Celsius. And what happens is these two strands come apart. They can't hydrogen bond together anymore. They come apart at high temperature, and then the reaction's cooled down to about 55 degrees C. And as you cool down, there are some primers. There's a forward primer and a reverse primer. And so they can actually bond again onto these molecules, strands. There's one for each strand. They face in opposite directions. They will bind to the strands, and then the Taq polymerase will fill in again the other strand, remaking again the strand. And so now what we have, and these will all be hydrogen bonded together, what we've done is we've gone from one copy to two copies. So these are two copies of the initial double-stranded DNA. So you've doubled the DNA. And the nice thing about PCR is that any one of these double-stranded DNAs is just like the first one. If you cycle back and go to 95 degrees, they will melt apart, we'll cool it down, primers will dock on, and you'll remake the strands. And so you end up making 4 double-stranded DNAs on the next cycle. So in this case, it would be cycle number 3. And then in cycle number 4, you get the idea. We are going to double things every time. So now we're up to 8, and then you'll go to 16 and 32, etc. And it turns out that even after just 20 cycles of this, so if you use your calculator and you put 2 to the 20th power in there, and that's multiplying 2 by 2 20 times, you'll find the number gets very, very, very large very, very quickly. And once you get a whole bunch of this DNA, which are exact copies of sequence that have initially been derived from the virus, and how that is controlled is these primers, these forward and reverse primers that are used for making this DNA. They're designed in their sequence to just match sequences in the target that you're interested in. So we use this method to analyze the expression of RNA in human retinal endothelial cells and blood vessels, if we're looking at genes that we study that regulate how blood vessels take care of the blood-retinal barrier. But in this case, the same technique is being used to just say, okay, let's specifically test for the presence of this COVID-19 RNA in a sample. And this is a PCR reaction. Then it turns out that in the test kit, there are really 3 reactions. So the viral genome, it's tiny, but it's long enough that they kind of design PCRs to target a few different regions. And so there's 3 PCR reactions called just N1, N2, and N3. And that's just to make sure that at least 1 or 2 of your reactions go, because when you're extracting RNA from people's samples, it'll be very variable. Sometimes you'll get more RNA from one patient's sample or less from another, and they also might have different amounts of virus in there. Somebody might have a little bit, somebody might be really sick and have a lot, and so you might have some variability in whether you get a positive test in the PCR. What also happens in this PCR is the real-time aspect. And if I made a graph of the concentration of the DNA, the double-stranded DNA as you're doing the PCR reactions with the cycle number, you basically, it looks something like this. It comes up for a time and then it levels off. And this represents the amount of DNA that you're making. Now, we want to be able to see this. And so what happens is to be able to see this in a real time as it's happening, and if we have a certain threshold of signal level, if something comes up in a reasonable amount of cycle numbers, say less than 40 cycles, and it pops up above a certain detection threshold, we would say it's positive for having that target sample. And the way you do this in a real-time PCR machine is that double-stranded DNA, when we have primers docking onto those strands to do the PCR reaction, in addition to the forward primer and the reverse primer that's going to help us copy each strand, each of the opposite strand to double the DNA, there's another special probe that is made to bind in the middle of the sequence. And this probe has a fluorescent molecule on it, and we'll just call it a fluorescence with a circle, and it has a quencher. And we can shine a certain spectrum of light onto this system in the tube, and what will happen is the Taq polymerase that starts on this primer and makes this strand, it polymerizes behind it, but if it runs into the back of another piece of this strand, it chops it up, and it continues making all the way through. And what happens is that releases this fluorescence molecule so it can get far away from the quencher molecule. Because normally what happens is light will activate this, it will start to fluoresce in a way, but the energy gets quenched by here, by this quencher molecule, so nothing happens, you see no fluorescence. But as you chop this off apart, you release this free and light can shine on it and it can give off fluorescence. And in this case it's a green fluorescence, and that green fluorescence is what is measured every cycle in the machine to see the reaction occurring. So every time you double the DNA, you chop up one more of these probes, you double the fluorescence. So that's what happens. And that's called real-time PCR. And so what we'll end up in our instrument is getting traces of PCR samples. And so somebody that's positive would have reactions that look like that. Somebody that's negative would just keep going for 40 cycles of PCR, and they would just not have any COVID virus in them. And what we also do is there's another PCR for the RNase-P gene, which is a human gene, and that's the control to make sure that we've actually got RNA extracted out of a patient's sample. If you did the extraction and the RNA extraction didn't work, you might think the patient's negative, but maybe they're positive, but the RNA extraction didn't work, so they look negative. What would warn you about that is one of the reactions we do will be to detect this piece of DNA, RNA DNA, from the human sample, from the cells in the human sample, and if that was a dashed line here, some of these patients might have that reaction. They should all go positive. So if one patient had nothing, had a flat line here for the COVID virus PCR, but they had a detection for the RNase-P, I would probably call that a negative test sample. That person does not have the virus, but these people would have the virus. And that's basically how the test works. And so we're going to set that up, and we'll show you some tubes and everything in the machine once we get that running. But this is the theory of how we PCR test.